Hey guys, welcome to the video on chapter 20. In this video guys, I have 4 goals for you. First, I want you guys to understand how scientists clone genes and use this in their research. Second, I want you guys to understand how we study the expression of genes. Third, I want you guys to understand how cloning organisms can be used in genetic research. And finally, I want you guys to understand how students will be able to explain how biotechnology is being applied to help us in our everyday lives. Well, I guess first the question has to be asked, what is biotechnology? This is basically learning how to manipulate organisms for research and practical purposes. A good key term to know here that goes along with this is the idea of recombinant DNA. This is DNA that comes from 2 different sources, and oftentimes it's the backbone of biotechnology. We take DNA from 2 different animals, or a plant and an animal, or 2 different bacteria, and we combine it together to create something new and unique. We call this entire purpose genetic engineering. So whenever we are actually changing the genes of an organism, we call this genetic engineering. So I guess the first question is, how do we study DNA? Well, the problem here is this. What if you want to know what kind of protein a gene creates? It's really kind of hard to figure this out if you think about it, because a cell is going to have a lot of different types of proteins, and there are lots of genes in every single cell. So how can scientists find out what a gene does, or what kind of protein a gene is going to create? And then once you have it, how can you isolate it and figure out what kind of protein is being made? Well, the first idea, guys, is an idea called gene cloning. What happens in gene cloning is this, is you have a bacteria that has a plasmid of some kind. Now remember, plasmids are small segments of DNA inside of bacteria. What then happens is that you can take a segment of a gene from some other source. So this can be from a human cell of some kind. And say you have a gene you want to study. You can then take that gene, put it onto the plasmid, and now you have a recombinant DNA plasmid. So you have a DNA that's recombinant because it comes from 2 sources, the bacteria and the cell of interest. You then have that bacteria can undergo mitosis, create a whole bunch of copies of itself, and now you have a whole bunch of copies of that gene. What you can do now is, if you want, you can pull the gene out and you have a whole bunch of copies of that DNA that you can now study to do some basic research on maybe what are the codes in that letter. Or you can look and see, well, what protein was created when we put that inside of the bacteria. The idea here is pretty simple. You take a gene from what you want to know about, you put it into a bacteria, and then you see what kind of protein is made as a result. Some key terms you have to know here, guys. And sorry, this whole process is called gene cloning, and it's basically producing multiple copies of a gene. It allows us to get multiple copies of DNA, and that will let us actually research and figure out what it does within an organism. A key idea here you kind of have to know, and we covered this earlier, but it's a big part here, is a plasmid. You must know that the DNA in this case is being inserted via a plasmid. It's not actually part of the bacterial chromosome, it's just a small segment of DNA that we are inserting into the bacteria. So then the question comes down to, how do you get the DNA that you want? So now you know that we can clone DNA to get the DNA we want, but how are we actually going to get DNA copies and to see what kind of proteins are made? But how do we know, like, what kind of DNA we're going to get? Well, the trick here is another thing called a restriction enzyme. And what a restriction enzyme is going to do is, it's going to take a section of DNA, and it's going to look for a series of letters, which is called a restriction site. Basically, the enzyme looks for a certain code of letters, just like this right here. And it knows whenever it sees this code of letters, it's going to cut it in a very obvious way, just like this right here. Once you have the cut, you now have a DNA that it's going to look kind of like this, where you have a DNA, and it's going to have a cut in it like this. What's going to happen next is you can take the DNA segment you want to study, the part of the gene you want to look at, and you can insert that into the gene, and you can then use ligase to fix it. What this then does is that using restriction enzymes, you can basically insert DNA into a chromosome. And a couple of key ideas here is restriction enzymes are basically just enzymes that cut up DNA and then can place new DNA inside that strand. A couple of key terms you have to know for this is restriction site. This is basically where the enzyme is going to be cutting. So that's right here is the restriction site right there. The restriction fragment. Well, the fragments are going to be, since it's cutting the DNA apart at these little restriction sites, usually the cells have more than one of these restriction sites. So the DNA, if you had a long strand of it, ends up being cut in several locations. We call these cuts restriction fragments. And that's the basic idea here. So if you want to cut open DNA and insert and remove strands, you can use restriction enzymes to do that. So one question we have here also is that if we have DNA, how can we store it for the long term? And that's what it comes down to, something called a bacterial artificial chromosome. Basically what you do is, if you have a gene, a series of genes you want to study from an animal or a plant or a human, you can cut those genes up, and then you can put each gene into its own individual bacteria, like this, and then you can make clones of it, and what you end up having is, you end up having bacteria that only have one gene. And you just label it and say, okay, this bacteria has gene 1, this bacteria has gene 2, this bacteria has gene 3, and then you can store the bacteria for later if you want to study what that gene does. Now each bacterial cell contains the gene that's stored in the plasmid, and you basically put it into a box that looks like this, and each one of these little containers here, the little circles, is going to store one gene. We call this a genome library, and basically a large collection of bacterial artificial chromosomes we call a genome library, because it's basically a library of DNA. So let's say you want to make more DNA. How can you do that? So you're at a crime scene, you found a small segment of DNA that you can study. What can you do next? What's the next goal? Well, you can use the situation here. If you don't have enough DNA to run tests with, well, you can make new DNA using a process called the polymerase chain reaction. It's kind of hard to read, but it's hard to get an entire graph of the polymerase chain reaction onto a PowerPoint slide. Basically, what a polymerase chain reaction does is it takes a sequence of DNA, so you have a strand of DNA inside of a test tube. You then heat the DNA so it's hot enough so the DNA splits in half. You then begin to cool it down, and you have primers and you have nucleotides inside the test tube, and as it cools down, what's going to happen is that the usual DNA replication process is going to take place, and it's going to make two brand new strands of DNA. So if you had one strand of DNA, you run the polymerase chain reaction once, you end up with two new strands of DNA. You run it again, you end with 4, you run it again, you end with 8. The end result here is you can basically create a whole bunch of DNA copies by basically running this process over and over again. So that's how we can get more DNA. Now, one other problem we have to deal with here in biology for biotechnology is restriction enzymes cut the DNA up. The problem is, if you have a long strand of DNA like this, and the enzymes might cut here, 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 here. It might cut it cuts in weird locations, so you might not get one easily even strand of cuts with restriction enzymes. So what you can do is you need to find a way to separate out these cuts, because this cut of DNA here, this might be 1, 2, 3, 4, and 5. You have different cuts of DNA here, 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 and here. What can then happen is you need to find a way to get this DNA and get it sorted out so that you don't have it all mixed together. Because if you want to study this strand, or this strand, or this strand, how do you get it separated from each other? Well, we use a technique called gel electrophoresis. What happens in this technique is it's a way to sort DNA. You use restriction enzymes to cut the DNA. So in the example up here, every single spot like this is a cut done by a restriction enzyme. And what's going to happen is that different strands of DNA are going to have different cuts, because you're going to have different locations for those restriction enzyme sites. And the result is you're going to get a situation where you have different size fragments. What you can then do is you can put these fragments of DNA into a gel electrophoresis machine, you can run it, And the end result is you're going to have the DNA sorted by size, where large fragments end up towards the front and smaller fragments end up towards the back. And if two fragments on this appear the same, then it means it's the exact same DNA strand because it was cut the exact same way. Basically, gel electrophoresis separates DNA by size. We will do this technique right here once we get back from after the AP exam. So we actually are going to do this, we're just not going to have time to do it before the AP exam. And that part I've already talked about. Okay, now at this point in time, guys, I've covered a lot of the major techniques, but there's a whole bunch of biotechnological techniques. I'm going to give you a brief summary of all of them, and that way you can get an idea of what scientists do. So one of the techniques is called the Southern blot test. This is basically a test to see where DNA is located. You can use a Southern blot test to find out if you want to find a certain gene inside of an organism, you can run a Southern blot test to find out if the segment of DNA you have has that gene. You can also use a technique called DNA sequencing. This is basically when you find out all of the letters in a strand of DNA. Basically, you can figure out the A, T, G's, and C's. This technique is pretty expensive, but it's getting a lot cheaper as we move forward. The Northern blot test is a lot like the Southern blot test, except where the Southern blot test looks for what DNA is being used, the Southern blot test figures out which RNA is being created. So if the Southern blot test determines if there is a certain gene present, the Northern blot test finds out what RNA is being created. So this here is a better test of gene regulation, and this test here is more about, is the gene present? The final one is called a DNA microarray assay. Basically, the DNA microarray assay is almost the exact same as Northern blot, except for Northern blot only tests for a couple of strands, so 1 or 2 strands of RNA. The DNA microarray tests for everything. It's like one big test to see what RNA is being created. So if this one here only looks for 1 or 2, this looks for all of them. The difference here is that the microarray is for known RNA samples. We have to know what RNA is present in the organism first. This here lets us investigate if the RNA is there. This tells us if the RNA is being created by a certain cell. There's one other thing called a genome-wide association study. Now, in a lot of animals, what we've done is we've basically taken DNA and we've killed part of it. So if we want to find out what a gene does, we oftentimes can take that gene in a mouse or another animal and we can kill it. And if we kill the gene so it doesn't work anymore, we can then see what happens to the animal as a result. The problem is that's pretty evil, and we don't like doing that because it usually kills the animal and doesn't always give us good results. So what we're doing now is something called a genome-wide association study. Because it's really easy to get a hold of DNA and sequence DNA now, what we're doing is we take a large number of people who have a certain trait, like let's say brown hair or blue eyes or heart disease or lung cancer. We then look at the DNA patterns. So we look at all of them, we look at all of their DNA, and we look for similarities. And we try to guess which gene controls for which trait. We basically look and see, okay, if every single person who has brown hair has these DNA sequences right here, then we can usually guess that those DNA sequences are used for making brown hair. And that's the idea here. We just look for lots of similarities. Another technique here, guys, is mostly pretty recent. It's called cloning. Cloning is basically making brand new animals. We take a copy and we make a copy of an organism. And it works like this. Basically, we remove a nucleus from the cell of the animal we want to clone. So we have an animal we want to clone, and we remove cells from it, and then we grow those cells in large number. Then we take a nucleus out of this cell and put that nucleus into the egg cell of a different animal of the same type. The nucleus that was inside this egg was removed by basically a giant tube. We go in, we suck out the nucleus. We then put the new nucleus inside the cell. The nucleus merges with the cell, and then bam, you get yourself a clone. And that's basically how this process works. It seems really easy. but it doesn't work very well right now. We don't fully understand the transcription factors involved, so oftentimes if we pull out the nucleus from the donor and put it into a new egg cell, oftentimes it doesn't work. Like, dogs are really hard to do this for, and the South Koreans are the best at it, and even they have to usually do it a couple hundred times before they get it to work right. So right now, it's mostly kind of a shotgun effect. They make this transfer process a few thousand times, and then hope one of them works. And usually one does. The problem is the other 999 don't work, and we don't know why it doesn't work. So this makes it look really simple, but understand, guys, we don't fully understand this process yet. Next idea here, guys, is called stem cells. Stem cells are a type of technology we've been using in which we basically, it's a cell that can become any type of cell. So a stem cell is able to become muscle cell, blood cells, nerve cells, cardiac cells, any cell you want. So we can find a way to control stem cells. We can basically regrow any part of the body that ends up being missing. It's kind of a cool little idea. But there's some problems here with this technology too. There are three types of stem cells. There's totipotent stem cells, which can become brand new people. Unfortunately, you only have totipotent stem cells when you are an embryo in your mother's womb. You don't have these kind of cells in your body anymore. They go away pretty quickly. Next kind of cell is called a pluripotent stem cell. These are stem cells that can become any kind of cell. However, they're also pretty rare, and by the time you're an adult or a teenager, all of these pluripotent stem cells are gone, so you don't have any more left. The last one's called a multipotent stem cell. This is a stem cell that can become pretty much any, it only can become a few types of cells. Oftentimes you have like skin stem cells that can only become skin cells. You have heart stem cells that only become heart cells. So basically they're stem cells that are limited in what they can do. So we have all this technology I've talked about. How can we use it? Well, I'm going to give you guys a brief overview of some of the ways this technology is being used. Well, first and foremost, we're using it in something called gene therapy. Gene therapy is when you try to treat a disease to give it some kind of new trait. Now, I say new trait because you don't just come about treating diseases, you can also change a person's DNA to give them new traits. Or as I like to call it, make me Wolverine. I would love to have the ability to be able to regenerate and do Wolverine-like stuff, so give me that gene. That might be possible here in the future. Right now, we're not super good at this. And largely it's because when we try to give genes to people, either it doesn't always work right, or we end up putting the gene in a bad location and cancer ends up resulting. So gene therapy is cool, but we usually only use it in situations where if there's no other option, because there's usually a high chance we might accidentally give the person cancer. Another type of situation is called transgenic animals. This is when we give genes to animals and have them make something we want. A good example here is this guy right here. Basically what we've done is we've taken goats and we've put a gene inside of goats that makes spider silk. The spider silk gene, because spider silk is super, super strong, so it would make a great product for clothes and for rope if you can find a way to make spider silk in large amounts. So they're trying to put spider silk genes inside of goats to get goats to make spider silk. Yes, it's creepy, but yes, it could work too. We call this a transgenic animal because we modified the animal to do something different. Here's some other cool stuff we can do. DNA fingerprinting. If you've watched any TV show in the last 10 years, you've heard about this. This is basically when you take the DNA of a criminal and find out, was it the person who did it? And it works based upon a technology called single tandem repeats. Basically, every single person has parts in their DNA which repeat with things like just T, 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 A, like this. Just random repeats like this. We call these short tandem repeats because they don't do anything. They just kind of sit there and act as barriers between genes. And every single person, because these tandem repeats don't do anything, they can mutate and there's no problem. Which means that most people have unique short tandem repeats. So here's an example. Let's say you have a murder victim or a rape victim. In this case here, we have 3 single tandem repeats. So the person here has 1700 on the first part, 1300 on the second part, and 1200 on the third part. So this is how many single tandem repeats they've got. Well, Earl Washington has a different number of single tandem repeats here, and Kenneth Tinsley has the exact same single tandem repeats. It's not brain surgery here. You can find out pretty quickly which one of these two people matches the victim, matches the killer. It's going to be Kenneth Tinsley because he has the exact same single tandem repeats as the sample they found on the semen. And that's how that works. You basically compare the number of single tandem repeats, and if they're the same, guess what? You found the killer. And that's it, guys. So this is a very, very broad overview of how this technology. I did not cover all the key terms. The vocabulary builder will cover everything else. And the activities that we're going to do later will help to show you guys how all this works as well. All right. Thank you very much for paying attention. I will see you guys.